Welcome to the Health Headliner podcast by Tudu Digital. Our content is informational only and not medical advice. Please consult your provider. Now, let's dive into today's headlines.
It's really incredible when you think about it. People living way up high in Tibet and Nepal, something like 90% of them share this a specific genetic tweak that helps them handle the super low oxygen up there.
It is amazing.
Yeah.
A powerful example of natural selection.
But then what really gets me is seeing something almost identical, but in a totally different place. People born with certain heart defects, so they have low oxygen all the time.
Right, chronic hypoxia.
Yeah, and they sometimes get these tumors. And again, it's like 90% of those tumors show changes in the exact same gene. How are those two things even connected?
Well, that's the big question, isn't it? And it's exactly what we're diving into today. We're looking at this really insightful study that digs into how our bodies cope with low oxygen, whether it's thin mountain air or the environment inside certain tumors.
Okay.
And we're basing this mostly on a detailed scientific paper, but also looking at a health news article that kind of puts it into context for us.
Got it. So the paper gives us the nitty-gritty, the genes, the cells, the patient data.
Exactly, the hard science.
And the news piece helps us see the bigger picture, why it matters.
Precisely. So our mission today, really, is to figure out the common threads in how the body adapts to scarce oxygen in these, well, seemingly unrelated scenarios, and especially to understand the really key role of one particular gene.
Okay, let's jump in. The study mentions this gene a lot, EPAS1. What exactly is that, and why is it so important when oxygen gets low?
Right, EPAS1. So that gene holds the blueprint for making a protein called hypoxia-inducible factor 2 alpha.
Hypoxia-inducible factor 2 alpha.
Okay. Bit of a mouthful. But think of it as a master switch for the body's response when oxygen is low system-wide. It kicks off those immediate things you might feel, like breathing faster, heart pumping harder.
Like when you go for a run or get startled.
Sort of, yeah. It triggers adrenaline and noradrenaline release, too.
Yeah.
And interestingly, it's also super important for newborns. They take that first breath and have to adapt from the womb to, well, breathing air.
Huh. Okay, so that's the quick response. But what about long-term low oxygen, like someone living their whole life up a mountain?
Well, that's where hypoxia-inducible factor 2 alpha plays another big role. When low oxygen persists, this protein, specifically in the kidneys, signals for more production of erythropoietin.
Erythropoietin, that's EPO, right? The hormone.
That's the one. EPO tells your bone marrow, hey, make more red blood cells. More red blood cells mean you can carry more oxygen. Super useful at high altitude.
Right. And the study says that in places like Tibet, among the Sherpas, there's been this really strong evolutionary push favoring certain versions of this EPAS1 gene. What does that actually mean for them?
It means that over millennia, individuals carrying specific variations in that EPAS1 gene were just better equipped to survive and reproduce in those high-altitude conditions. So those gene versions became incredibly common there, much more so than in lowland populations. It's a clear sign they offered a big advantage.
Okay, an advantage.
But here's the twist. The versions that became common often seem to slightly dampen down that hypoxia-inducible factor 2 alpha erythropoietin pathway.
Wait, hang on. If they need more oxygen carrying capacity, why would a version that reduces the EPO response be better? That seems backwards.
It does seem counterintuitive at first, doesn't it? But it's all about finding the right balance. Yes, more red blood cells carry more oxygen, but too many makes your blood thick, like sludge.
Oh, right. Harder to pump around.
Exactly. It puts a huge strain on the heart and can actually impair blood flow to tissues. So these specific EPAS1 variants probably help Highlanders avoid that too much of a good thing problem. They maintain enough red blood cells, but not so many that it causes other issues. It's like fine-tuning the system.
Okay, that makes sense. It's about optimization, not just maximization. The study also mentioned where these useful EPAS1 versions might have come from. Something about Denisovans?
Yes, the Denisovans. They were an archaic human group, ancient relatives who lived in Asia, including areas near the Tibetan Plateau tens of thousands of years ago.
And we know modern humans interbred with them.
We do. The genetic evidence is clear. And what's absolutely remarkable is that the specific version of EPS1 found in, 90% of Tibetans and Sherpas looks almost identical to the EPAS1 found in Denisovan fossils. But you barely find it anywhere else in the world.
Wow. So they got this adaptation from interbreeding.
It strongly suggests that, yes, this beneficial gene variant was likely passed on from Denisovans and then became super common in Highlanders because it conferred such a survival advantage in that specific environment. It's a really cool example of ancient mixing helping modern humans adapt.
That's fascinating. Okay, so that's the high-altitude adaptation story. Now, let's switch gears back to those tumors. The study looked at pheochromocytomas and paragangliomas.
Let's call them PPGLs, maybe. Bit easier.
Good idea, PPGLs. And specifically in patients with congenital cyanotic heart disease, so low oxygen from birth because of their heart condition. What did they find there?
Well, this is where it gets really striking. They looked at how often mutations occurred in that same EPAS1 gene within the PPGL tumors. And it depended massively on whether the patient had that underlying chronic low oxygen condition.
How different?
Hugely different. In patients with the congenital cyanotic heart disease, almost 89% of their PPGL tumors had mutations in EPAS1. 89%.
89. Okay.
But in PPGL, patients without that chronic low oxygen, only about 4.5% had EPAS1 mutations.
Whoa, 4.5% versus nearly 90. That's a massive difference.
It really is. Almost a 20-fold increase. It points very strongly towards the chronic low oxygen environment itself, somehow driving or selecting for these mutations within the tumor cells.
It has to be connected. Did they look at how strong that selection pressure was?
They did. They ran what's called a selection analysis on the tumor genomes from the patients with the heart disease. And sure enough, EPS1 popped up as the gene under the strongest positive selection, particularly in tumors coming from sympathetic tissues, parts of the nervous system that are really sensitive to oxygen levels.
And less selection pressure in the tumors from people with normal oxygen.
Much, much weaker signal for EPS1 selection in those tumors. It really isolates the effect of that chronic systemic hypoxia.
Okay. And what about when these mutations happen? Early on in the tumor's life or later?
That's another key piece. They looked at the fraction of cancer cells within a tumor that actually carried the EPAS1 mutation. And often it was very high.
Meaning it was present in most of the tumor cells.
Exactly. Which suggests that the EPS1 mutation is often one of the first things to happen, an early event that gives those initial cancer cells a survival edge in that low oxygen setting.
Right. So let me see if I've got this straight. We have this gene, EPAS, vital for handling low oxygen. In high-altitude folks, specific versions of it are super common, selected over ages to help them live there.
Correct. Inherited adaptation.
And now, in these PPGL tumors growing in people with chronic low oxygen, we see a huge number of new mutations popping up in that exact same gene.
That's the core finding.
It's like the tumor cells themselves are adapting to their own little low oxygen world.
That's a great way to put it. The study really hammers home this incredible parallel. You've got roughly 90% prevalence of EPAS1 alterations, the inherited variants in Highlanders, the somatic mutations in these specific tumors in both scenarios.
90% both.
It really suggests a fundamental shared biological playbook for dealing with low oxygen, even though one is evolution over millennia affecting a whole population, and the other is happening, much faster within the cells of a tumor in one person's lifetime.
And the researchers think this tumor adaptation might be sped up. Like evolution on fast forward.
That's the idea, yes. The constant, intense pressure of low oxygen might force these tumor cells to evolve rapidly, strongly favoring any change like an EPAS1 mutation that helps them survive and grow in that hostile environment. It mirrors the long-term process, but on an accelerated timescale.
Okay, but how? How does the low oxygen actually cause these mutations in the tumors? Does it damage the DNA or something?
That seems to be a major part of it. They did experiments in the lab using PPGL-derived cells. When they exposed these cells to low oxygen for extended periods,
What happened?
they saw more signs of DNA damage. And crucially, they saw lower levels of a key DNA repair protein called PARP1.
PRP1, like part of the cell's DNA fix-it crew.
Exactly. So the picture emerging is that chronic low oxygen can, 1, cause more damage to the DNA, and 2, hobble the cell's ability to repair that damage.
Yeah.
That naturally increases the chances of mutations occurring randomly across the genome, including in genes like EPS1.
Makes the DNA unstable.
Right. And there's another layer, too, something called oncometabolites.
Oncometabolites, cancer metabolites.
Sort of. They're metabolic byproducts in cancer cells that can actually fuel the cancer process. The study found that these EPAS1 mutated PPGL tumors had higher levels of one called fumarate and lower levels of another called succinate.
And these affect DNA repair, too.
Apparently so. Changes in these metabolites, which are linked to how the cell makes energy, can also interfere with DNA repair pathways.
Hmm.
So it's like a double whammy, more damage happening, less effective repair, and metabolic changes hindering repair even further.
Wow, it really does sound like a perfect storm for mutations inside those tumors.
It does, and they didn't stop there.
Mm.
They also looked at how these EPAS1 mutated cells were actually using oxygen, focusing on the mitochondria, the cell's power plants.
Did they use more oxygen? Less?
Surprisingly less. The cells with the EPAS1 mutations showed lower rates of oxygen consumption. They also found these cells had ramped up production of an unusual version of a key energy-making protein component, cytochrome C oxidase subunit 4 isoform 2. Let's maybe call it COX4I2.
Okay, COX4I2. So less oxygen use, more COX4I2. What?
The thinking is, it might be a strategy to be more efficient with the limited oxygen available, rather than just trying to burn through it as fast as possible, optimizing use when the resource is scarce.
Huh. Like sipping fuel instead of guzzling it. That's clever, in a way. And this connects back to early development somehow. You mentioned neural crest cells.
Yes, exactly. PPGLs originate from these neural crest cells, which are present early in development and form tissues like the ones that sense oxygen. The researchers found that during normal development, EPAS1 and this COX4I2 are expressed together in these immature cells.
But not usually in adults.
Right. Their expression typically goes way down after birth. But in these EPAS1 mutated tumors, that coordinated expression pattern seems to reappear. It's like the tumor cells are reverting to an earlier, perhaps more hypoxia-tolerant developmental state to help them cope.
A kind of cellular time travel for survival. This is all incredibly complex. Did they connect this back to actual patient outcomes? Like, did the level of oxygen really correlate with getting these tumors?
They did. They analyzed data from a large international group of patients, and they found a clear, strong link between having congenital cyanotic heart disease and developing PPGLs.
Supporting the hypoxia connection.
Definitely.
Mm-hmm.
And more specifically, among those heart disease patients, the ones who did develop PPGLs generally had lower blood oxygen levels.
Lower than other patients with the same heart condition.
Yes. And they were also less likely to have had early surgery to fully repair their heart defect, surgery that would have restored more normal oxygen levels sooner.
That seems like pretty strong clinical evidence linking the chronic low oxygen to the tumor development.
It really does. It ties the lab findings nicely to the clinical reality.
And there was something about the timing of the low oxygen influencing the type of PPGL.
That's another interesting observation from the data. It seems that if the low oxygen is present right from birth congenital hypoxia, it tends to favor PPGLs arising from those sympathetic nervous system tissues we mentioned earlier. But if the significant low oxygen starts later, maybe in childhood or adulthood, it seems more associated with parasympathetic PPGLs, specifically tumors in the carotid body and the neck, which is a major oxygen sensor.
So when the hypoxia hits matters for where the tumor starts.
It seems plausible, yeah. It might relate to how developed those different tissues are at different life stages and how they respond to low oxygen then.
Okay, so stepping back. This study is really bridging these two worlds. Long-term evolution at high altitudes and rapid tumor adaptation in low oxygen. What are the really big takeaways here?
I think one of the biggest is this idea of evolutionary convergence. Seeing the same gene, EPAS1, pop up as central to adaptation in such different contexts, generations of Highlanders and individual tumors, it really highlights a fundamental biological response to low oxygen. Life finds a way, often using similar tools.
A shared solution to a shared problem.
Exactly. And for cancer specifically, it really underscores how chronic low oxygen can be a potent driver of tumor evolution. It's not just a passive bystander. It actively shapes the tumor by causing DNA damage, hindering repair, and selecting for mutations like EPAS1 that promote survival in that environment.
Which suggests low oxygen could be a factor in more cancers than we think.
Potentially, yes, especially in the core of solid tumors, where oxygen supply is often poor. This research definitely elevates the importance of the hypoxic microenvironment.
And does this open up new treatment avenues?
Oh, absolutely. Since hypoxia-inducible factor 2 alpha seems so critical for these EPAS1 mutated tumors, drugs that block its activity look very promising. There's one called belzutifan, for instance, that's already in clinical trials for PPGLs.
How's it looking?
Early results seem encouraging shrinking tumors, reducing hormone levels in some patients. It's still early days, but it's a direct therapeutic strategy based on this biology. Plus, the DNA repair angle.
With the PRP1 protein being reduced.
Right. That might make these tumors vulnerable to drugs called PRP inhibitors, which are already used in other cancers with DNA repair defects. So maybe a combination approach could be effective down the line.
It's just wild to think that studying people on mountainsides could lead to targeted cancer drugs.
It really shows you the power of basic science and looking for connections across different biological systems. The researchers even suggest that studying how organisms adapt to other stresses like intense sunlight or scarce food might reveal similar vulnerabilities in other cancers.
Fascinating. Now, we should probably touch on limitations. No study is perfect, right? What are the caveats here?
That's always important, yes. Well, first, as we discussed, this study really honed in on one specific tumor type, PPGLs, and one specific cause of chronic hypoxia, that congenital heart disease.
So we can't necessarily say this applies to, say, lung cancer or hypoxia caused by poor circulation.
Not directly, no. We need more research to see how broadly these specific EPAS1-driven mechanisms apply to other cancers and other hypoxic situations. It's likely complex and context-dependent.
Okay, that's fair. What else?
Well, the lab experiments using cells grown in dishes, while super informative for mechanisms, they don't perfectly capture the full complexity of a tumor growing inside a person, with all the interactions with blood vessels, immune cells, and so on.
The real-world environment is messier.
It is. And finally, while the links are strong, teasing apart the exact interplay, the precise contribution of the underlying heart condition's genetics versus the hypoxia itself, versus the DNA damage and repair issues leading specifically to EPAS1 mutations that still needs more detailed investigation. It's a web of factors.
Right, the full sequence of events isn't perfectly mapped yet.
Exactly. It's a significant step forward, but the complete picture is still developing.
So if you were summarizing the expert take on this, what would be the headline? What's the real significance here?
I think experts would be really struck by the elegant connection drawn between evolution on a grand scale and evolution within a tumor. That convergent evolution story around EPAS1 is powerful. It reinforces fundamental biological principles.
Bridging evolution and cancer biology.
Precisely. And then there's the clear clinical relevance. Identifying hypoxia-inducible factor 2 alpha as a key driver and potential drug target for these specific, often difficult-to-treat tumors is a major practical outcome. It offers real hope for patients.
So both fundamental insight and clinical potential.
Absolutely. But, seasoned researchers would also add that necessary note of caution. This is complex. We need more research, especially to understand how these findings translate to other cancer types and hypoxic conditions. It's a launchpad for more work.
It really does make you wonder, though, if our bodies have these incredibly intricate ways to adapt to oxygen levels, something so basic, yet it can be life-saving in one scenario and maybe fuel disease in another, what other hidden adaptations are going on in response to other chronic stresses, or even just inside a tumor's chaotic world?
That's a great question to ponder.
This study feels like it's opened a window onto that adaptability, from our genes right down to our cells. It makes you think, what other unexpected connections are waiting to be found if we keep looking at biology through this adaptation lens?
It's a really exciting perspective, isn't it? It shows the depth and sometimes the surprising logic behind biological processes. If this deep dive sparked your interest, I definitely encourage exploring more about high-altitude physiology or the role hypoxia plays across different cancers. That EPAS1 gene and its protein, hypoxia-inducible factor 2 alpha, they're really hot topics now in understanding and treating certain diseases. It's a field moving fast, always more to learn.
Thanks for tuning in to the Health Headliner podcast by To Digital. Like and subscribe for more health news and breakthroughs. Stay healthy and informed. See you next time.